And about total leukocyte count, okay, means total WBC count, right? In our blood cells there are three type of blood cells: the red blood cells, basically the red blood corpuscles, the WBCs and the platelets, isn't it? You people have already done the total RBC count. Today we are going to learn about WBC count, isn't it? So what is the total leukocyte count of our body? It is basically the number of cells that is present in 1 cubic mm of blood. Okay, 1 cubic mm of blood of a person contains this much number of WBCs is the total WBC count. Okay, or the total leukocyte count. Okay. Also 1 cubic mm can be expressed as 1 microliter of blood. Okay, and this blood is basically an undiluted blood. The blood that you take from a patient, this blood contains this much number of cells in every 1 microliter or 1 cubic mm of blood. Okay, so this is the total leukocyte count. Now there are some normal ranges of total leukocyte count. Okay, the adult normal range of total leukocyte count is 4,000 to 11,000. Okay, this 4,000 to 11,000 is the normal WBC count or the total leukocyte count of an adult person. Okay, now you can see in case of a child at birth there is cell count of 18,000 plus minus 8,000. Okay, so basically it is at a increased number compared to all the adults, isn't it? So there is some reason to that. This having WBCs count much more or much higher than adult, there is some reason to that. The main reason behind is but you basically know from your higher secondary studies that in our body there are something called immunoglobulins, isn't it? There is IgG, there is IgM. In an adult, basically all the immunoglobulins are present, okay, to fight any sort of infection. Now in case of a child, what happens after birth, it takes few months, mostly 6 months, to develop all the types of immunoglobulins. And you also know that placenta cannot transfer all types of immunoglobulins from a mother's blood to a fetal blood, isn't it? Because of the size and some other restrictions are there, isn't it? So to fight any sort of infection after a baby is born, baby requires some other protective measures. This protective measure is the increased WBC count, isn't it? WBCs also fight infections, isn't it? So this is a protective measure for a baby. There is also increased hematopoiesis is there. So in hematopoiesis there is total RBC count also increases, WBC count also increases. Okay, there is increased bone marrow activity. With time what happens is, with age your bone marrow activity reduces. Okay, the bone marrow activity reduces, and hence you can see at 1 year the WBC count comes down to 6,000 to 15,000. At 2 to 6 year it comes down to 5,000 to 15,000, and at 6 to 12 year it comes down to 5,000 to 13,000, which is almost like an adult count. Okay, adult total WBC count. Now in case of a pregnancy, the total WBC count increases. Okay, because in pregnancy what happens, there is a fetus inside mother's womb, isn't it? Now mother's body has to do much more activity to protect herself and even the fetus to fight any sort of infection. Okay, so there is increased production of WBCs in mother's blood in pregnancy. Okay, so now what was the total count? 4,000 to 11,000, isn't it? So any WBC count which is beyond 11,000 is called leukocytosis, and any WBC count which is less than 4,000 is called leukocytopenia or leukopenia. Okay, so what are the causes of leukocytosis? Now this leukocytosis basically happens not just due to some pathological cause but also in physiological cases also. Okay, for example, in case of exercise, what happens in exercise? You tend to do more work than what you do in a resting state or in a daily state, isn't it? Now when you do more work, there is increased production of adrenaline in your body. There is more and more adrenaline rush in your body into the blood, and also there is increased amount of and hormone known as cortisol. Okay, you know something that is called cortisol. Okay, so now due to cortisol and adrenaline, what happens is there is increased WBC release into the blood. There are two things basically happen. One thing is there is increased release of WBCs into the blood. One more thing is there. Basically there is a single layered endothelium around our blood vessel, isn't it? Inside our blood vessel there is single layered endothelium. What happens is WBCs are basically staked with the layer of the endothelium. Okay, there are WBCs in the circulation, then there are also WBCs that are sticked with the endothelial layer. Now what happens is in case of increased flow, when you do exercise, what happens? Your heart rate increases, isn't it? Your blood pressure also increases. There is increased blood flow. It causes what? There is shifting of these WBCs from this sticky endothelium into the circulation. Okay, so there are two things happening. One is WBCs are releasing from the bone marrows because of increased amount of cortisol and adrenaline. Next thing is the WBCs are released from the endothelium that were stick to the endothelium. Okay. At but physiological cause I have explained to you what is the reason behind that. Then there is muscular exercise, there is pregnancy, I have told you, there is high temperature. What happens in high temperature? This is basically comes under physiological. You know, high temperature means fever. Fever basically comes to some pathological causes. So any pathological causes, for example infections, in most cases of infection what we find is there is fever. So your body tends to basically fight those infections by increasing the amount of WBCs in our blood. Okay. Now in physiological causes the same thing happens. In physiological causes, if someone has fever without any infection, there is also increased amount of adrenaline in your blood. Okay, so these do what? These do the same thing. They basically releases WBCs more from the bone marrow and also releases WBCs from the endothelium that is stick to it. Okay. Then there are severe pain. Also in case of severe pain there is leukocytosis. Now comes the pathological causes. In pathological causes there are infection, there is malignancy called leukemia, and then there are severe hemorrhage. Infections, their body tends to fight infection, there is increased production of WBCs, isn't it? In case of malignancy, there is a malignancy called leukemia, which in general term is called blood cancer, isn't it? Now in case of leukemia, there is basically leukocytosis which goes beyond 50,000 per microliter of blood. 50,000 cells per microliter of blood. This is called leukemia. Then there is severe hemorrhage. What will happen in case of a severe hemorrhage? Your body will tend to compensate for that, isn't it? There is loss of blood, there is huge loss of blood. In severe hemorrhage, body will try to compensate by increasing the production of all the blood cells in the bone marrows, isn't it? Now in severe hemorrhage, when the bone marrow is basically hyperactive to form more and more amount of RBCs, in the same bone marrow only WBCs are also produced. You know, there is red bone marrow, there is white bone marrow, isn't it? Both comes under bone marrow only. So there is hyperactive bone marrow. This hyperactive bone marrow basically does what? They increases the level of RBC to compensate for severe hemorrhage, and in that case only there is increased amount of WBC also because of hyperactive bone marrow, that is the white bone marrow also. Okay. So there are leukocytosis in case of infections, malignancy like leukemia and severe hemorrhage. Okay, this comes under pathological classification. Okay, severe hemorrhage, there is loss of blood from our body. Okay, so body tries to compensate for whatever has been lost. What will happen? There will be hyperactivity of our bone marrow to increase more amount of RBCs. These are basically lost by severe hemorrhage, isn't it? So when there is hyperactivity of bone marrow, there is also the production of WBCs. As we know, RBCs are produced from red bone marrow, WBCs are produced from white bone marrow. So during hyperactivity of bone marrow, there will be increased number of RBCs as well as WBCs also. Okay, so this is the reason why there is leukocytosis in case of severe hemorrhage. Okay, about leukocytosis, the physiological causes and also the pathological causes.
Exercise because I want you you listen carefully, try to learn all these points carefully so that you can answer these questions during your exam biology or in theory. It is for you.
So now we are shifting to leukopenia. What is leukopenia? Leukopenia is reduced WBC is less than 4000 per. You have to always say the unit, okay? It will be asked in your exam. Always you have to mention the units. Less than 4000 cells per cubic mm or microliter of blood, okay? So what are the causes of leukopenia? One thing is bacterial infections. Like what do you know that earlier I have told, in case of infections there is increased amount of WBCs, isn't it? It should increase. By our logic, WBC should increase in case of infection. But what we are seeing here is there is reduced WBC count in case of typhoid. Okay, what happens in typhoid is there is this typhoid organisms, basically the cytokine mediated there is a cytokine mediated depressed activity of the bone marrow in case of typhoid infection, okay, or the enteric fever. So there is reduced amount of WBC in case of typhoid infection. There comes is hepatitis and influenza under viral infections. Okay, in viral infections like hepatitis and influenza, there can be two things. One thing is the virus directly going into the bone marrow and they are suppressing the bone marrow activity. There is one thing. The second thing is, in case of viral infection, in case of any infection, basically there is some sort of inflammation, isn't it? There is some sort of inflammation. There are some sort of markers are associated with the inflammation. Now what happens is, in case of viral infections like influenza or hepatitis, some sort of markers in those viral infections basically target the bone marrow cells, and what happens for this reason there is reduced activity of the bone marrow. Okay, so there are two causes, basically virus directly acting on the bone marrow and suppressing it, and then there are inflammatory mediators that are basically going there and suppressing the bone marrows. Okay, then there are protozoal infection like malaria. What happens in malaria? There is hepatomegaly, there is splenomegaly. In case of splenomegaly, what happens is all the lymphocytes there, in case of splenomegaly, they are basically, when the spleen increases, there is increased amount of filtering of the lymphocytes. When increased amount of filtering occurs in the lymphocyte, most of the lymphocytes get stuck into the spleen. So there is less number of lymphocyte into the blood. As a result, we know lymphocyte is the second most contributor of WBC count into our blood. The first is the neutrophil. It comprises of almost 60 to 65%. Then there is lymphocyte. So when there is lymphocyte count reduces, there is reduced WBC count. Okay. Then there is megaloblastic anemia. Do you know what is the cause of megaloblastic anemia? Can anyone tell me why megaloblastic anemia occurs? Iron deficiency causes iron deficiency anemia. Megaloblastic anemia, anyone? Folic acid deficiency. One more thing is there, vitamin B12 deficiency. Folic acid and vitamin B12. Now do you know what is the basically the action, the function of folic acid in our human body? Huh? Something else is there in our cellular level. What happens? What does folic acid does? Folic acid is involved in formation of folic acid is involved in biochemistry, that DNA synthesis has been over or not? Not, I guess. Okay. So basically folic acid is involved in formation of DNA. Okay. Now when there will be decreased level of folic acid, in case of megaloblastic anemia, you will find there is reduced amount of formation of WBCs. Okay, since the folate level is less, there is less amount of DNA that is formed, so there is reduced amount of WBCs also. WBCs has nucleus, isn't it? Every nucleus contains what? DNA. So when there is reduced amount of folate, there is reduced amount of DNA, so there is reduced amount of WBCs in case of megaloblastic anemia, not in case of iron deficiency anemia, I am telling you. Okay. Then there are some causes that lead to bone marrow depression like aplastic anemia. Okay, aplastic anemia is a sort of anemia where there is depression of the bone marrow that causes reduction in number of total WBC count, total RBC count, and also the platelet count. All the blood cells basically decreases in case of aplastic anemia. Okay, then there are some drugs like chemotherapeutic agents are there. They causes reduction in number of WBC count by depressing the bone marrow, and then there is radiation. Okay, so these are the causes of reduction in WBC count or leukopenia. Right? Clear, everyone? Typhoid. In the typhoid, basically what happens, there is same way, there is depression of the bone marrow. This infection causes depression of the bone marrow, okay, in enteric fever.
So
Malaria in case there is splenomegaly, isn't it? Spleen basically does what? They basically destroy the RBCs. Then there is also another function of spleen is they filter out the lymphocytes. Lymphocytes are formed in, there is maturation of in lymph nodes, but the filtering of the lymphocytes occur in spleen. So when there will be splenomegaly, in case of malaria, there is increased filtering of the lymphocytes. So maximum amount of lymphocytes get stuck into spleen. And you know spleen lymphocyte is the second most contributor of WBC count, isn't it? So there is reduction of the number of WBC. Basically reduction in number of lymphocytes, okay, contributing to leukopenia. Written, everyone has noted it down. Megaloblastic anemia is basically folate is less. I have told you now, megaloblastic anemia basically occurs due to reduction in number of folate or there is vitamin B12 deficiency. Now folate is involved in DNA synthesis. Since there is reduced amount of folate, there is reduced amount of DNA synthesis. Now WBCs are nucleated cells. You know na WBC, what is the difference between WBC and RBC, anyone? Non-nucleated. So what is the full form of RBC? Corpuscle, because they loses nucleus at their maturation stage. Okay, same way WBC doesn't lose any nucleus, they have nucleus. Once they have nucleus, that means that there is presence of DNA also, isn't it? So what happens is, in case of megaloblastic anemia, there is reduced amount of folate in our blood, so there is reduced synthesis of DNA, so there is reduced amount of WBCs also. Okay. So we are going to learn about total leukocyte count, how to estimate total leukocyte count. There are two methods, okay. What we do here is visual hemocytometer method, that is by using the Newborn's chamber, okay. Then there is one more method called the electronic method. You just have to know about it. You don't we don't practice it here in our lab, okay. As all of you must have attended the RBC count, the class of the total RBC count. So you know what is hemocytometer, isn't it? Hemocytometer. Cells in the blood that is counted by some slide in the chamber, that is the Newborn's chamber. Okay, there is this Newborn's slide, there is this cover slip that forms a 3D space called the Newborn's chamber. Okay. Now in case of total leukocyte count, you use two types of blood. Either you use capillary blood by pricking into your finger, isn't it? What you will be doing in your exam is pricking your finger. Now in case of our practical class, we are doing what? We are basically taking blood from your vein and mixing it with EDTA in a vial, isn't it? And we are providing to everyone so that you can practice more and more. But in case of exam, what you have to do is you have to prick your finger, you have to take blood from your finger only. No EDTA blood will be provided to you, okay? So you take two types of blood. One is EDTA blood, that is blood in the vial. You can see purple colored vial is there. This is the EDTA blood. Then there is capillary blood that you can take by pricking your finger, okay? Or full form of EDTA, anyone? And what does it do? How does it function? Chelating what? Chelating what? What is the thing that is involved in your coagulation cascade? Huh? Not platelets. Only calcium is involved in coagulation, nothing else is involved. Na? Among the ions, only calcium is involved. So it is chelating the calcium so that the coagulation cascade cannot happen and the blood doesn't coagulate. That's the way how an anticoagulant works. Okay. So the requirements are microscope, hemocytometer, and there is WBC diluting fluid. In case of RBC count, we have learnt about RBC diluting fluid. There are two types of fluid. Now this is WBC diluting fluid. And in case of total leukocyte count, what you use is a different type of pipette. This is known as WBC pipette. Okay. This WBC pipette has basically the grading of 0.5, then 1 before the bulb, and then after the bulb it is 11. In case of RBC count or the RBC pipette, the pipette was having grading of 0.5, 1, then 101, isn't it? So, and another thing of marking the differentiation is there is white color bead in the bulb. In case of RBC pipette, there will be a red color bead. Okay. So what do you need? Newburgh slide, cover slip, and then WBC pipette, not the RBC pipette. So you know all these things, right? Newburgh chamber, there is Newburgh slide, putting the cover slip, it becomes a Newburgh chamber. The depth is 0.1 mm. There is an H-shaped groove or the gutter area where the excess fluid gets drained, isn't it? You people know, right? So this thing have I already shown basically. So see, in case of RBC count, we are considering this large box, isn't it? What is this name of this large box? This large box? Or Newburgh chamber is the whole thing, na? What is the name of this square? Huh? How is it small? Then which one is the large square? Anyone? What is the name of this square? RBC square. What type of square is it? Is it a small square, smallest square, or large square? Large square, no? This is a large square. Every large square containing how many? There are 25 small squares. And every small square containing 16 smallest squares. You have considered this central large square in case of RBC count. Now in case of WBC count, we will be considering the corner large squares. There are 4 large squares in the 4 corners. We will be considering these 4 large squares in case of total WBC count. Okay. So we have shown you already in the class that the dimension of this Newburgh chamber is basically 3 mm into 3 mm, isn't it? So what will be the dimension of this area of this large square? 1 mm into 1 mm. So if we tell you what will be the volume of the diluted fluid in this area, once you put the cover slip, what will be the volume of this square? What will be the volume of this square? 1 mm into 1 mm into?
What is the height of the chamber?
What was the depth or height of the chamber? 0.1 mm. So it will be 0.1 cubic mm. The volume of the fluid will be 0.1 cubic mm. So we can give them some exercise.
I'll give you some problem solving. You will expect some questions in theory as well as in practical. These are problem solving. This is one of the examples of this is now unit 5. You better. What is the length from here to here? 3 mm. Sure? Okay. It is written. Good. Okay. Then what is the length from here to here? Means it is square. Okay. You have divided it into how many parts? So what is the length of each part? Be sure. 0.25 or 0.025? 0.25. So 0.25. What is the area of this area of this square? Area. 1 mm. 1 mm. Mm square. Okay. So sir was again today also say was asking you, always tell us the unit. So area is 1 mm square. Now what is the volume of this area? Zero point 1 cubic mm. Okay. What is the volume of this area? Okay. Everybody now able to calculate the area, volume? Sure, but he is not sure. Volume of this.
Of this square, right?
Right. 16 divisions. 16 small. So I divided it by 16. Okay. And I got the average value for each small square. And the value is 2. The value is 2. Okay, the value is 5. The value is how much? 5. Now you know the volume of this small square?
Yes.
Now can you calculate how many WBC will be there in 1 cubic millimeter? Do it. मान लो blood is not diluted. Blood is not diluted. मान लो. बाद में मैं बोलूँगा. तो अभी क्या है? One small square is having 5 WBC. In 1 cubic millimeter, how many WBCs will be there? Calculate. You calculate how it can become 80. You calculate. It's not so easy. In 1 cubic millimeter. निकालो देखो दिखाओ कैलकुलेट करके देखो You got my problem? Huh? one smallest square is having 5 WBCs. Now I know the volume. Now I want to know what would be the value for 1 cubic millimeter. The number of WBC here, this is the upper left corner. There is another corner here, upper right corner. There is another corner here, lower right corner. I can see another corner there. There is lower left corner. So you have to count all these 4 chambers, corners. How many? How many smallest squares are there? Total, in total. 64. One is having 16. So number of 16 into 4. So you will be counting number of WBC in all these 4. So what is the area? This much WBC will be having in this area. What is this area? This is the area. You know the area now. So this area contains this mass of WBC. Say, what should I say? What is the value to be given? Say total 1, 2, 3, 4. 160. Total value is 160. Now, all. Now, what is the value per cubic millimeter? Millimeter.
The number of WBCs are 160.
Among these 4 in total.
All the 4 quadrants. Yes.
You have to count 4 corners, no? So total WBC is 160. You know the volume of all these 4 corners? 4 quadrants, you know the value? So this volume contains 160. How much? It is very simple. Now you are ready. If you can do it, now you are ready for the problems. Sir, give whatever problems you want to give us, give.
WBC Pipette. I have told you what is the differentiating marks for WBC Pipette and RBC Pipette, isn't it? There is a white colored bead in a WBC Pipette, okay? This is a WBC Pipette. There is a white colored bead in the bulb of a WBC Pipette. In case of RBC Pipette, there is this red colored bead. The markings or the gradings are 0.5, 1 and then 11 in case of WBC Pipette. In case of RBC Pipette, it is 0.5, 1, 101, okay? And the graduations is up to mark 11 and there is white colored bead, okay? Now in next slide you will see what we do is basically we take blood the same way. In case of RBC count, we take blood till 0.5 mark, then we take WBC diluting fluid till 11, okay? Now what does this WBC diluting fluid comprises of? There is one thing called glacial acetic acid. The concentrated acetic acid is there. Then there is 1% gentian violet. Then there is distilled water 97 ml. Okay. What do you mean by that 1% gentian violet, 1% white by volume? Can anyone tell? What does it mean? 1% white by volume means? Not 1000. 1 gram in 100, not 1000. Okay. What is the function of the glacial acetic acid? Basically, when you take blood, you have known, right? You have known to in other class that the WBC is to RBC ratio is 1:700, isn't it? That is, the RBC count is very high. So if we take blood and we do cell count, what we will see under microscope is RBC only. We are not going to see WBC because there will be so much amount of RBCs. Means every 700 RBCs there will be only 1 WBC, isn't it? Because the count is very high of RBC. Now what we do is we use glacial acetic acid. What does glacial acetic acid do? They basically lyse the cells. through the lipid layer of the cell membrane, they enters into the cell and they do lysis of the cell. Now in case of RBC, since this RBC doesn't contain any nucleus, once the cell is lysed, nothing will come out of this RBC. No nucleus is there, so it will be completely invisible once the RBC is lysed. The same way the WBC membrane can also get lysed, but what happens is we are basically seeing the nucleus of the WBCs. You know there is nucleus in WBCs, okay? Now to see the nucleus of the WBC, we are using gentian violet. This gentian violet basically stains the WBC nucleus. Only the nucleus, not the whole cell. Because the cell get lysed, only the nucleus is present, and this nucleus can be seen by staining with gentian violet. So once the all RBCs are destroyed, all the layers of the WBCs are also destroyed, only thing left is nucleus which are stained by gentian violet. Okay, then there is 97 ml of distilled water which is a solvent. Okay, so see, the glacial acetic acid lies the red cells while the gentian violet slightly stains the nuclei of the leukocytes. Blood specimen is diluted till 1:20. What was the dilution in RBC count, anyone? 101 is the grading. What is the what was the dilution? 200 dilution. How did it came to 200? You took 0.5 part of blood, then you took fluid till 101. That was, and in first part, in the before the bulb, this grading till 1, whatever the fluid was there, this basically didn't get mixed with the blood you took. So 0.5 part blood was present in 100 parts of fluid, the RBC diluting fluid, and hence the dilution factor was 0.5:100 or 1:200. It's the same way in WBC pipette, when you take blood till 0.5 and then you take dilution fluid till 11, there is 0.5 part of the blood present in only 10 part of diluting fluid, because the same way this one part of the fluid is basically not mixing with the blood. And what you do before charging, discard 2 to 3 drops, so that you discard the unmixed fluid from the pipette before charging. Okay, so the same way this 0.5 part of the blood is basically mixing with 10 part of the fluid. Okay, and hence the dilution factor comes up to be 0.5:10 or 1:20. Okay, so after that what you do? You do the counting. Now in case of counting, the same thing, only it has been written. In case of counting, the same way you do is, there are 4 large squares. For 4 WBC squares, every WBC or every large square contains 16 small squares. What you need to do is, the same way you have done in RBC count, you have to count it as zigzag method, and also you have to use this left and lower formula. Okay, any cells touching the upper border or the right border you can't consider. You only consider the cells touching in the left and the lower border. Okay, and you have to count it in zigzag way, so that you don't count a cell twice. Okay, zigzag fashion you have to do in every large square. That is, ultimately at the end of your experiment you have to count total 64 small squares. Every small square, large square containing 16 small squares. So you have to count all the 64 squares and then do the multiplication. Okay, see, can anyone tell me how many cells are there? यहाँ पे कितने cells है? That you will be counting, not total cells. 19. 18 किसका 18 है result? किसके 18 है? Is it 19? 18 किसका आ रहा है बोलो? तुम्हारा. और anyone? Anyone else contributing to 18? 18, isn't it? Or anyone? Anyone else? कितना आ रहा है यहाँ पे? How much? 16 और 19 और किसी का 17 आ रहा है? 17 और 20 नहीं आ रहा है. 19 आ गया, 18 आ गया, 16 आ गया. isn't it? हाँ 17, yes. Next 18 चाहिए मुझे. 18 किसी को आ रहा है? No, 18 तो आया है, इसका है. Okay. So the cell count is 19. You have to use your eyes only, nothing else. In this case, you don't need to use your brain. You only use your eyes. See, the cells that shouldn't be counted has been given cross, isn't it? The cells that shouldn't be counted has been given cross already. The right one, the upper one, isn't it? So what cells are you counting? Only the cells that are not being crossed. And there are 19 cells actually. Not 18, not 16, or anyone have told not 17 also. It's 19. So, in 16, what will be the WBC count? Or WBC count in 1 cubic mm, what will be the count? Total ko to ashbe. What will be the total count here? If there are 19 cells in 16 squares, कितना आएगा? 190. 190, isn't it? 19 cells in 0.1 cubic mm, isn't it? What will be the volume of this square? 0.1 cubic mm. So 0.1 cubic mm contains 19 cells, 1 cubic mm, 190 cells. Now the thing is, the same way in case of RBC count, you also have to do multiply it by dilution factor. Okay? So let us see how the calculation is done. See, this is the calculation. I have told you, right? You have to count cells in all the large squares. There are 4 large squares. So basically this N represent total cell count present in all the large squares, or all the 64 smallest squares, small squares. There are no smallest square. So all the 64 small squares, okay, or all the 4 large squares, the total number of cells, this is the N, okay. Now you are multiplying it by dilution factor of 20 and dividing it by 4 into 0.1. How does it come? Basically 0.1 cubic mm is the volume of 1 large square, 0.4 cubic mm is the volume of 4 large square. So basically 0.4 cubic mm contains n number of cells, 1 cubic mm contains n by 0.4 number of cells, and since you have diluted the blood till 20, it comes up to be n by 0.4 into 20. Very simple, okay? And finally it comes up to be n into 50. But try to remember the formula, okay? Because in your practical exam, basically this will be some diverted. It will not be like dilution factor हम लोगों ने 20 दिया है. We can change the dilution factor, isn't it? We will give, for example, consider dilution factor of 30. Then you have to use the maths by using the formula. Okay, do the maths by using the formula, right? There is another method at the beginning. I have told you, electronic method. This electronic method is done by how? This, you take the blood, you just give it into a cassette is there, you introduce the cassette in the machine, this machine will automatically count the cells, WBCs, RBCs, platelets, everything. How do you confirm this type of electronic method reading? You do confirm by visual hemocytometer method, because the visual hemocytometer method is the standard method. Okay, what you will be doing, right? So there are some advantage and disadvantage to this electronic method. It's easy and rapid, time saving, large number of cells can be counted, and high level of precision. The disadvantages comes to costly calibration error. Nucleated RBCs are counted as leukocytes. This is a very important point. The nucleated RBCs are counted as leukocytes. Are the RBCs nucleated? No, right? RBCs can be nucleated. Sometimes the premature RBCs get released into the blood. For example, I have told you, in case of severe hemorrhage, what will happen? Increased release of blood cell from the bone marrow. So in case of severe hemorrhage, even before the maturation takes place, the cells will be released into your blood. Okay, so you will see premature RBCs, that is nucleated RBCs, in the blood also. Okay, so nucleated RBCs are counted as leukocytes. That gives you a increased leukocytosis while the patient is not having any leukocytosis. Okay, and then there can be platelet clumps also. This platelet clumps also gets counted as leukocytes in electronic method. That's why you compare it with visual hemocytometer method. Okay, that is the standard method. Sir, I am basically finished.
This is how to do
45 डिग्री में देखा अभी कैसा हुआ है. That's how you do it. कैसे करना होता है? You have to touch it in the edge of the cover slip, not into the edge of the pipette. Okay? ये रख दो. You can use it. ये चार्ज हो गया. यहाँ पे थोड़ा एक्स्ट्रा फ्लूइड है ना? Give me some tissue paper. So एक्स्ट्रा वाला फ्लूइड कैसे सोक करोगे? You have charged it. Let it settle. Let the WBC settle down till 5 minutes. After 5 minutes, what you do is you just touch the surface of the cover slip. Whatever the extra fluid is there over the cover slip, it will get soaked. See, it is getting soaked. Don't touch this part because then the fluid from the chamber will come and soak on it. Now you have to keep it till 5 minutes and then we will see on the microscope. Bring it till 11, then immediately make it horizontal. Or what you can do is keep the pipette in your mouth only and make it horizontal. Make it horizontal. Okay, now discard the extra part. Don't tilt it. अभी एक्स्ट्रा पार्ट डिस्कार्ड करो ना. किसी को काउंट नहीं करना. डिस्कार्ड कैसे करना होता है? यू डोंट नो. देखो remove your hand. When you draw the fluid, see how to discard. You have to keep an eye on your pipette, okay? So that it comes to 11. Draw any excess fluid. Please come to 11 ना. अभी ऐसे काम में रखना है और 2 to 3 मिनट मिक्स करना है. ऐसा नहीं करना है. ऐसे करोगे तो इट विल गो to the center of the video. Because ऐसे रखना. You have to keep it horizontal, okay? You have done the RBC count, isn't it? Okay, so I will show you na. Do it. First do the mixing. 2 to 3 minutes, okay?
इधर कर दिया. सुस्मत क्या है? क्या ये ठीक होगा?
Reflect.
कितना हो रहा है वो लेंगे मेरा
इसका क्या पर्सनल ये है क्या ना
इसमें देखिए क्या हुआ इसका हुआ
चेक कर रहे हैं
हाँ चेक कीजिए